Hi, everybody. I wanted to provide you guys with some information about the lab you guys will be completing today or during this week, which is on leaf litter ecology. So the lab activity that is associated with this PowerPoint is already available to you. So you guys may take a look at it anytime. Please do note that this lab activity has a due date on Canvas, so keep an eye on that, please. So when we're talking about leaf litter or basically any environment that may have a cycle of life, we will be dealing with what we call decomposers. And basically, decomposers are either small or large organisms. It could be even microorganisms that are responsible for doing the final breakdown of our organic matter. So, for instance, you start with a leaf, it breaks down to the most essential components of that leaf, and it can be recycled and used to produce other components. Now, there is quite a few types of decomposers in our environment. They include bacteria and fungi, which I think some of you guys may already be familiar with. But we also have mites, beetles, centipedes, fly ants, wasps. All of these are identified as decomposers and many other. If you guys take a look at your actual activity, we do have a list of these decomposers in more detail. Now, for today, what we're looking at is we're looking at four different type of leaf litter that was collected underneath four different trees. So basically, our goal is to see how the organisms that are actually completing the decomposing process differ from one leaf litter to another. Now, these organisms that are basically carrying out the process of decompositions have been distributed in their environment either based on the specific adaptation that they have, so certain characteristics, genetic components that makes a specific leaf litter a nice and habitable environment for them. And they also, again, those specific adaptations help them survive in a very specific environment that they can tolerate. So how can we use the information that we have here to basically complete our lab today, which I think is actually a very cool lab. So basically what we do is under normal circumstance, we would go outside and we will identify four environment or four leaf litter within four different type of environments. After identification of these environments, which we actually identify as microenvironments, we will collect a sample. And the way we collect the sample is we basically we're using these rings that goes on the surface of ground, and we collect any leaf that is falls within the scope of that ring. It basically gives us a same sample size for all of the leaf litters that we're collecting. And again, the goal is to eventually look at the organisms that are found within these leaf litters and see how they work together and what is the relationship or interrelationship between them that allows them to accomplish their job. So again, the procedure is very simple. Usually it happens over a few days. We first of all, again, as I mentioned earlier, we choose a specific location. We want to collect our leaf litter, and we use what we call, again, a sampling ring, which is simply, think about it, a ring about 22 centimeter in diameter. All we do that is to put that ring on a surface, and anything that falls within that scope of that ring will be picked up and put in a Ziploc bag for collection. Now, each of these samples are going to be taken back to our lab and be placed in a separate setup that you can see on your slide. So basically, this setup is like this. You have a funnel, and the funnel, right on top of it, you have a sieve, which basically allows things to pass through. The end of the funnel is set into a ethanol solution, which is basically alcohol. And right on top of this space, we have a light. So the idea is that as these insects kind of start to move around within their leaf litter, they're gonna pass through the sieve and fall into the ethanol, and that would be our collected sample. Now, typically, again, we can do this in 1 to 2 days, but because of your lab, typically it extends about 2 weeks. The first day for the collection and the second day for analysis of the organism that has been collected. So some of the examples of decomposers, I just want to show you some pictures. So you have your kind of your roly-poly, your slug, a snail, millipede, earthworm, which are usually not observed unless you have really thick leaf litter or it's been raining. You have a springtail, you have mites, you have a lot of ants. Here's other examples of what you can see as and identify as decomposers. You have this kind of, again, your mites, your termites, snails. In the larger aspect of it, you have your centipedes, your millipedes, your earwigs, spiders, flies, wasps, beetles, ants, etc. So the idea here is, again, if you were in a class and you were completing this lab, each group would receive one of these samples that we have collected at the bottom of all of our funnels, and you would look at these guidelines that is provided here and identify each individual decomposer. Now, I mean, you don't get a chance to do that in class and in person. So what I've done is actually look at 4 different leaf litter for you and collected the data for you, and I'm basically presenting that data to you. So to kind of give you an idea where these samples are collected, one was collected underneath a sycamore tree, one underneath a passion fruit tree, which also has a lot of leaf litter right about now. Same thing as avocado trees, and then honeysuckle. So the idea is, again, as we look at these leaf litters, they may have the same organisms that are your decomposers. However, the frequency of these might be different. So here is the 4 samples that I basically collected data for. So you have your sycamore tree, your passion fruit, avocado tree, and then honeysuckle. You have the species that are basically observed, and then the frequency of those samples or each of the specimens. Now notice that there are variation, of course, between them. Some have relatively small numbers, some have quite a few. So the idea here is, again, you guys are going to use this data that was collected for you to do some data analysis and answer a few questions. So what is the first thing that you want to do? The first thing we want to do is identify the frequency of each of these, or percentage of each of these specimens within their own individual environment. For instance, if you look at the ants for the sycamore tree leaf litter, we have 15 ants. And the total insect data that was collected, if you add them up all together, it would be about 37. So if I'm doing the calculation to see what's the percentage of ants that are found underneath the sycamore tree, I take 15 and divide it by the total number of insects, which are 37, multiply that by 100, and that gives me about 40.5%. And you can round them up to one decimal place for your calculation. So basically what you do is, if you go forward, I actually provided these charts for you as part of your lab activity. So you go to your charts and you say 40.5% of the samples under the sycamore tree would be ants. And then you repeat the same calculations for these. So you say centipedes are 4, total numbers are 37. So 4 divided by 37 times 100 gives you the frequency or the percentage. Now you have to do this for every single component right here and the other 2 samples. So then again, total of 4 data tables you guys need to complete. So that's the first part of the lab. The second part, you guys are going to graph this data. So feel free to graph it by hand, or if you're comfortable using Excel, go ahead and do that. So what you're doing again, you're doing 4 graphs. On the x-axis, you have the name of the animals. So let's say again, ants, mites, wasps, etc. And then the percentage you calculated on the previous slides. So make sure that each of the datas are properly labeled as an, okay, this is the data or graph for sycamore trees, this is for passion fruit, this is for avocado, etc. Now, the last thing you guys are going to do is you're going to do a calculation that shows you the percentage of each insect in all of the total insects you have collected. So let's go back and I'll just kind of show you what I mean by this. Sorry, back. So right here, if I'm looking at ants, I have 15 ants on this sample, 35 on this sample, 50 on this sample, and 21 in this sample. So you have 21 and 50, which is 71, 71, 81, 86, and then 105. So, oh, sorry, 115. So you have 115 ants in all of the samples you collected. Now you're going to divide it by the total number of all the insects from these 4 tables. So basically total this, total this, total this, and this, add them up all together. And then you're dividing the total number of ants by the total number of the insects. So let's say you had 50 ants, and, oh, sorry, let's say you have 115 ants, and all of the insects you collected were 300. So you write 115 divided by 300, and then multiply that by 100. This is going to help you guys basically answer one of the first questions you have in the handout or the activity. Now, after you guys done your calculation and figure out your percentages, all you need to do is basically go back to your activity and answer some of the lab questions. I believe there's 3 questions. Just FYI again, this is the lab that will be due online next Tuesday by 11:59 p.m. All you need to do is download the lab activity, type in your answers, complete the charts directly into the lab activity, and then make sure you load it up online. The graphs can be done either by hand and submitted separately as part of your report, or if you are really savvy with Excel, you can make them on Excel and add them as part of your lab report or lab activity. As always, if you guys do have any questions or concerns, please let me know, and I will see you next week.